Okay, excellent. Can everyone hear me okay? Good, excellent, fantastic. Oh wait, I have to share my screen. That's why we practice this. Okay, just make sure I can see. Okay, hopefully now you can also see my screen and you're hearing me. Excellent. Okay. Well, yeah, I'm delighted to be here today. I've come to know just a little bit about SSP and what a cool program it is. So, yeah, I'm happy to have this opportunity. So, my name is Rebecca. I'm an associate professor at the University of Guelph. We're like an hour outside of Toronto or so, for those in not Canada. And the research in my lab is focused on studying the genetics primarily of fungal pathogens. So we're going to start talking a little bit about fungi and then dive into kind of some of the work that we do. And these are actually just some of the yeasts that we work with. Let me get my inner thing. Oops. Did that work? No. Okay, never mind. It's the wrong thing. This. Oh, none of that's going to work. Okay, never mind. We don't need that. Okay, so there we go. Okay, so yeah, I want to start talking about a bit about fungi since that's sort of the topic of all the work that we do. So fungi represent an entire kingdom of life. So they are incredibly diverse, right? So to be differentiated from animals or plants, which are also entire kingdoms to themselves. So fungi are an entire kingdom of life and they're incredibly diverse and they perform all sorts of critical functions in our world. They are responsible for allowing most plant species to live and thrive. They are important decomposers in the environment. They produce critical metabolites. They produce things like antibiotics, statin drugs that are used to lower cholesterol are produced by fungi, all sorts of incredible functions. But they're also in kind of the right context and the right organisms can be really critical pathogens. And fungi are frequently known as kind of important pathogens of insects, also important pathogens of amphibians, including frogs and other related species. You may have seen headlines about kind of massive frog extinctions that are caused by a devastating fungal disease. Fungi are also really prevalent and important pathogens of plants. So far more than viruses or bacteria, plants are deeply affected by fungal pathogens. And so this is responsible for kind of massive die-offs of important crops. And so that has obviously important implications for our food security. Again, if you see, when you see headlines about things like extinctions of like banana species or things like that, those are caused by fungal pathogens. And then relevant to the work that we're interested in my lab and something that I think is of interest and concern to many is that fungi can also be pathogens of humans. And I'll talk about this in a little bit more depth. I also always feel obliged to sort of highlight now because we were not really primed previously to think about fungi as pathogens of humans until in the last couple of years when a blockbuster HBO show came out that made everyone suddenly really concerned about the potential of kind of human-associated fungal disease. So this was, The Last of Us, which merges the idea of these sort of cordyceps fungi, which are real pathogens of insects, with the idea of kind of human fungal disease. Now this is very much not true, and we don't have to worry about this, but just a good thing to think about. Okay, so when we are actually thinking about fungi as pathogens of humans, we're often thinking about two sort of broad categories. So fungi are very frequently found in infections that are fairly superficial to the human body. So this includes common things like what we often call yeast infections, vaginal yeast infections. Things like athlete's foot are caused by kind of skin-dwelling fungal pathogens. Ringworm is kind of a misnomer, but is caused, in fact, by a fungus. and even dandruff, which of course is incredibly prevalent. Maybe we don't think of it as a disease per se, but dandruff is also caused by fungi. And so these kinds of infections, again, they're fairly superficial, right? They happen on the skin or the mucosa. And while they tend to be fairly easily treatable, they are incredibly common. So they impact, millions or billions potentially of people every year. On the other side of the coin are fungal diseases that are far less common, but much, much, much more difficult to treat and which can be life-threatening. And so these are caused by three sort of major groups of fungi. There are others, but these are kind of the ones that cause by far the most a number of kind of invasive life-threatening infections. And so these include Aspergillus fumigatus mold, which is inhaled from the environment and can cause lung infection, so like a fungal pneumonia. Cryptococcus neoformans, which is a yeast fungus that can also be inhaled from the environment and cause fungal pneumonia, and can also disseminate to the central nervous system and cause fungal meningitis, which can be very difficult to treat. And Candida pathogens, which are actually the ones that we focus on in my lab, and are one of the most common causative agents of both kind of superficial fungal infections like vaginal yeast infections, but can also cause invasive life-threatening infections. And I'm highlighting here because we don't tend to think about fungi as being this sort of important cause of human infectious disease. And when we look at this graph on the top, this is looking at the estimated number of global annual infections from some sort of well-known infectious disease agents, right? So malaria, which is a parasite, tuberculosis, a bacteria, HIV, AIDS, obviously a virus. And when we think about invasive fungal infections, the number of infections is vanishingly small compared to other sort of more common infections globally. But when we look at the number of estimated annual deaths caused by these types of infections, people are often surprised, to see this data where invasive fungal disease is responsible for about as many deaths globally every year as things like AIDS or tuberculosis, despite the fact that there are, relatively very few of these infections. And so this tells us that the mortality rate associated with them is quite high. So while they can be rare infections, they can cause very, very serious disease. I won't go into this sort of in too much detail, but these tend to cause infections in people who are severely immunocompromised, which is why we don't see them as prevalently as something like malaria, for instance. And just to highlight also that just, I think maybe 2 or 3 years ago, for the first time ever, the WHO sort of finally recognized the really growing importance of fungal infectious diseases and highlighting fungi as sort of priority pathogens for more research. And so in my lab, what we're interested in studying primarily are Candida pathogens, right? So that was one of those kind of 3 major kind of classes of fungi. And Candida are interesting. They are yeast species, so they grow in this sort of single-celled form. This is what they look like growing on a petri dish. But some of them are polymorphic, so they can grow both in this sort of yeast morphology, but they can also grow as these elongated filamentous cells and these hyphae. And when we kind of report to we look at the WHO critical fungal priority pathogen list, we know that Candida species are some of the most common and sort of most critical organisms in this group. And so most of the work that we do in my lab focuses on Candida albicans. This is by far the most common Candida species, but there's many other kind of critical Candida organisms. And in particular, I will highlight here Candida auris, which I will talk about again in a second, which many people may also have heard of. As Candida auris is a highly, highly drug-resistant pathogen, so resistant to almost all the antifungal drugs that we have, and has been a sort of newly emerged cause of fungal disease. So it's a pathogen that actually didn't exist or was not causing infections about 20 or less than 20 years ago. So it's really kind of a new emerging pathogen. And so, when we think about these difficult to treat infections, we often, of course, have to, reflect on how it is that we can treat them. And so the development of new antifungal drugs to treat invasive fungal infection has been quite limited, and there's a lot of challenges with the development of new antifungals. First, it's important to note that compared to the number of classes of antibiotic drugs, right, so things that are used to treat bacterial infections, where we have maybe at least a dozen or so sort of very broad classes of antibiotics, we only have about three types of antifungals, right? So we're really limited in the kinds of drugs that we have to treat these infections, which again is why we have such high mortality rates, which can rise to as high as about 50% for these invasive infections. And so there's a lot of challenges with antifungal drug development. It's costly. Companies aren't kind of often willing to invest in this. We get a high rate of evolution of antifungal drug resistance. This is something I think people are familiar with in the case of antibiotics, right? We talk about antibiotic drug resistance and these kind of antibiotic, resistant superbugs. So we have the same thing with antifungal drug resistance, right? These pathogens become kind of able to tolerate the treatment with these compounds. And the other kind of major concern when it comes to fungi, which is distinct from the development of antibiotics, is that fungi actually share a really remarkably close evolutionary history with the humans that they infect. So on the sort of broader tree of life, bacteria are prokaryotes, right? They're like very distantly related, but both fungi and humans are eukaryotic cell or made up of eukaryotic cells, which on the kind of grand scheme of evolution are remarkably similar. In fact, in kind of molecular biology research, we often use Saccharomyces yeast. We use a yeast species like bread beer yeast as a model organism to study human biology. That's how similar they are, right? So trying to develop a drug that will hurt or kill a fungal cell without hurting or killing the human in which it resides can be a really difficult challenge. And I just want to zoom in particular on the concern of antifungal drug resistance, right? Again, this idea that these pathogens sort of evolve to resist treatment with these drugs. I said early on that fungi are not just pathogens of humans, they're also incredibly common pathogens of plants, right? So we spray antifungals in the environment to try to prevent infections on crops. And these maps are showing over the last couple of decades the kind of number of different strains of species identified to be resistant to antifungals that are either plant pathogens or human pathogens. And you can see that over the last couple of decades, we've seen from what was pretty much kind of a non-existent problem to a really large number of species of both crop and animal fungal pathogens that are highly resistant to the drugs that we're trying to use to inhibit them, which is a problem. And just to zoom in on a couple of some of the specific challenges that are associated with this, because some of this will kind of flow into the work that we're doing. So one is that which I touched on a second ago, is the rapid rise of some newly emergent multi-drug resistant pathogens. And so this again gets us back to Candida auris, which I think, again, many people have seen in headlines in the last couple of years. This was a species that was not even identified until 2009, I think. So it didn't even exist. And then less than 20 years ago, one infection with this organism was found. And then since then, this has been the rate of the number of infections of Candida auris bringing us to 2022. So what went from 0 to about 2,000 infections about 5 years ago. And that may not seem like a large number of infections, but this is a brand new pathogen that never existed before. And it is highly, highly, highly resistant to basically all antifungal drugs, right? So this is highly problematic. Another important concern with fungi, because fungal infections tend to overwhelmingly impact people who have compromised immune systems, it means that, for instance, in the case of the COVID-19 pandemic, this was also seen with the AIDS epidemic, when people are quite sick with another disease, be it AIDS or COVID-19, we see the co-occurrence of fungal disease. And so during the COVID-19 pandemic, for instance, we saw a surge in both Candida auris infections and also something called mucormycosis, which is otherwise a quite rare fungal pathogen, but which caused devastating sort of smaller epidemics alongside the COVID-19 pandemic. Again, as a result of all these kind of quite sick and hospitalized people. Another major concern with fungi, I touched on this idea that we use antifungals both in the clinic and in agriculture, right, to inhibit the growth of fungus on crops. And one of the major problems that we have is that one of the classes of antifungals that is the most used, which are the azole-based antifungals, have been used more and more in agriculture in the past years. So this graph is showing basically the amount of azole antifungals that are being sprayed on crops from the early 90s until 2016 is the latest data. And I think, it doesn't take much to read this chart to see that we have skyrocketing rates of spraying these azole antifungals on crops. And what more recent sort of genomic analysis has shown us is that fungi in the environment that are being sprayed with these antifungals, they develop resistance to them in the environment. And then when human patients are exposed to these fungi through, soil and the environment, they can develop a fungal infection, which is already resistant to the antifungals that we would use to treat it in the clinic. And that's because the fungi have been exposed to those antifungals in the environment. Right? So you can imagine how this is a problem. And then the final thing to touch on, of course, is climate change. So some fungi have only exist in certain regions, right? So they grow in sort of soil or different environments, and they have a particular niche that they like to grow in. And there's a lot of evidence and projections that show that as the climate warms, certain fungi that have had a more restricted geographical niche are going to start expanding. We've seen it already, and the projections show that it will increase. And that we might see just the emergence of sort of new species of fungi, like we have already seen with Candida auris, that we might start seeing kind of more organisms start to appear as pathogens of humans. So this is sort of the context in which we are about disease. And so in my lab, and this is like obviously a huge broad topic. There are a lot of people who are interested in this biology. And so in my lab, we're interested in understanding human-associated fungal pathogens. And we're interested in a couple of different aspects of this. One is that we're interested in understanding the ways in which they become resistant to antifungals. So we're interested in looking at the kinds of mutations and things that occur that take what would be, a susceptible strain to become a resistant strain. And I'll talk about some of this work. We're also interested in developing new genetic manipulation techniques. We work mostly with CRISPR editing that allow us to manipulate the genomes of these fungi in order to understand what some of the genes are that are involved in resistance. And then finally, we're also interested in the kind of identification and development of new antifungal drugs, because I think, hopefully it's clear that as resistance becomes a problem, we need to make sure we have sort of continual new kind of types of antifungals around. And so I'm going to tell you sort of two vignettes of work in my lab that touch on these themes. So the first gets at this kind of the last thing I mentioned, which is the development of new antifungals. And so what we've been doing here is a machine learning based approach towards antifungal drug discovery. And so when we think about the discovery of antifungals or any sort of antimicrobial in general, like an antibiotic as well, what this usually relies on is looking at existing libraries of chemical compounds and screening them in the lab to identify ones that can inhibit the growth of a fungus or whatever organism you're interested in. And this has limitations, as you can imagine, right? So we have, compounds, we incubate our fungi with them, we let them grow, and we see which ones are inhibited by those compounds. So the limitations of this are I mean, it's very manual. It is sort of limited in scope. Into like how many compounds you can actually kind of realistically screen. It tends to have a very low hit rate, right? So you can screen, tens of thousands of compounds and identify only a few that have activity. And it tends to be quite biased because the kinds of compounds that we have that we can screen tend to already have biased activity towards certain functions. So we're not as kind of likely to necessarily identify a new antifungal. There's a lot of limitations to this approach. And so this brought us sort of the idea of using machine learning for antimicrobial discovery. Now, we are not the only ones taking this idea of using machine learning for new drug development, but this is, to our knowledge at least, the first kind of application of it for antifungal drug discovery specifically. But the idea, sort of in a more general approach, is that what we can do, I think all kind of have a sense now that machine learning at a very simple level takes some sort of training data as its input and uses kind of complex models to generate output that we can then kind of validate and interpret. And so machine learning sort of predict or computationally identifies patterns in these large complex data sets. And that allows us to make some predictions about other related data. And so what we're trying to do is sort of use this model where we feed in known antifungal drugs. And what we hope to kind of come out of our output is predicted new antifungal drugs, right? So can we train kind of these models to predict what makes a good antifungal drug? And again, this is an approach that certainly is being used extensively for antibiotics, other types of therapeutics, and this is the kind of application of it for antifungal drug discovery. And all of this work that I'm going to talk about is actually a collaboration with another Canadian lab, John Stokes, McMaster University, who himself has been applying machine learning models for antibiotic drug discovery. And so we're kind of now turning this into an antifungal project. So the first thing we've been doing here is using a discriminative machine learning model for antifungal drug discovery. And what that allows us to do is to screen a relatively small number of antifungal compounds on the order of a couple of thousand, not of antifungal compounds, to screen a smaller number of compounds for antifungal activity. So in this, we screen about a couple of thousand drugs to see which ones have antifungal activity. We then feed this becomes our training data, right? Which of these drugs has antifungal activity? We feed this training data into our ML models to train it. And then we can use this to analyze much, much larger sets of molecules on the order of about 30 billion molecules from computer databases to then predict which of these looks like it would be an antifungal and which of these looks like it would be not an antifungal. It's sort of a binary output based on what it has learned from the training data, which is actual kind of screening in the lab. And so this is work that's been pioneered by a couple of awesome grad students in my lab, Eileen Bates and Mia Fogel. And they did exactly this. So they screened about, this come up, they screened about 10,000 chemical compounds in the lab. And then they looked for the ability of those compounds to inhibit the growth of the Candida fungi that we work with. And basically in blue here are our kind of non-hits, the ones that allow for growth of the Candida as shown here. And the red ones, which fall below this line, are ones that inhibit the growth, because this is growth on this axis here. And so these are the ones that have antifungal activity, right? So from this, we now have kind of our training data of non-antifungals in blue and red, the actual ones that have antifungal activity. And so again, we feed this into our model and we use this initially to predict the activity from a chemical database, like a computer database of about 12 million chemical compounds. And so we asked, of these 12 million compounds that are on a computer, which of them look like they have antifungal activity and which don't, based again on what we know from this training data. And so we feed in these 12 million molecules with our training data. From it, we get an output of how many compounds the model thinks will have antifungal activity. And then from there, we manually do a bunch more refinement. So we try to eliminate anything from that list that is already known to have antifungal activity, because that's not interesting. We're trying to find new antifungals. We get rid of anything that we think is going to have toxicity to humans, right? Because we don't just want a poison, right? We want something that's going to specifically kill fungi. We look at the cost, a whole other kind of set of things. And this allowed us to initially prioritize 35 compounds of interest. And so what we did from here is we bought these compounds. They're ones that are sort of readily commercially available. And we looked for their ability to inhibit the growth of our Candida pathogens. And what we're looking at here is just two different concentrations of, or actually 35. Did I say 35? Anyways, 35 compounds, along with some controls. These are existing antifungal drugs, fluconazole and caspofungin. And in green, it shows a fungus growing really well. And in black, it's showing a fungus basically dead and the spectrum in between. And so what you can see is that we've, from this, identified 3 compounds, 19, 21, and 35, that had pretty good antifungal activity, again, compared to our sort of antifungal controls. So this might seem low. This is about 3 out of 35, but that's about, a 10% hit rate, which is just like orders of magnitude higher than what we would expect if we were just kind of screening compounds. So from this, we've identified these sort of 3 new antifungal agents. We're following up on 2 of them, 19 and 35, as we're calling them. This is just showing the amount of drug that is needed to inhibit them. So we do like a whole range of dilutions of the drug to find out how much is needed. We're excited about these because actually only a very, very tiny amount of the compound, about 0.5 micrograms per mil, is needed to inhibit the growth of the fungus. And that's great because you don't want to be using drugs at like super high concentrations. You want something that's very potent, and that's what these are. And so we also, we have also looked at how effective these compounds are against other organisms. So all of our screening was done in Candida albicans, but we've looked at how well they can inhibit other Candida species, including Candida auris, right? That's that sort of scary multi-drug resistant emerging pathogen, as well as some distantly related pathogens like Cryptococcus, which is a very, very kind of distinct fungal pathogen. And this is looking also at the growth across different concentrations of drug. And with the exception of this one Candida krusei strain, we find that both the compounds, I'm only showing one of them here, are highly, highly effective at killing almost all the fungi that we have tested them against. So that's great. It means it has fairly broad spectrum of activity. Whoops. Okay. And so we're doing sort of in-depth follow-up on these compounds. I'm not going to go into that in too much detail, but I do want to highlight the sort of next arm of this work that we're working on, which is using, instead of a discriminative, using a generative machine learning model to sort of facilitate our next generation of antifungal drug discovery. And what happens with a generative model is the model builds totally novel, previously non-existing molecules from chemical building blocks. And so we use very similar training data, but instead what the model does is it takes these sort of small little chemical units. We have about 130,000 chemical building blocks, these sort of small molecular units that can be combined by 13 different reactions, yielding about 50 billion potential molecules. And we do this. So we sort of start with a single, I'm pointing to my screen, I realize you can't see that. We start with a single building block, and then we add on different molecules in different combinations through these reactions. And again, are the generative model that interfaces with the previous model that I described to then allow us to distinguish which of these, novel compounds would be a good antifungal or not. We also do this in a way that predicts compounds that can, in fact, be generated, right? So they don't exist kind of currently. You can't buy them right now, but they can, theoretically at least, be synthesized in a lab. So this is the kind of next generation of how we're approaching this. And I don't have kind of data to show you on this yet, but we have predicted a bunch of new antifungals in this way, and we're in the process of getting them synthesized. So we're excited to see how that goes. Okay, and so that was sort of like the first, slightly more than half of kind of our research that I wanted to talk about. And the second part is looking at CRISPR tools that we're using to understand antifungal drug resistance. So instead of thinking about new antifungals, here we're thinking about, with the existing antifungals that we have, we know that resistance develops quite quickly, and we want to understand that's happening. And so this takes us to sort of an interesting phenomenon that happens in fungi, which is that, so we know that they can become resistant to antifungals quite quickly. And one thing that we know is that sometimes fungal pathogens like the Candida that we study, they actually hold on to an extra copy of some of their chromosomes. And this makes them more resistant to antifungals. So the Candida that we work with is a diploid organism, just like we are. It means we have 2 copies of all of our chromosomes, or it does, and so do we. And when an extra copy appears, we call it a trisomy, right? So there should be 2, but instead there's 3 copies of a chromosome. And in fungi, it turns out this happens quite frequently. They become trisomic quite often. And some of these trisomies, these extra chromosomes that I'm showing here, make the cell more resistant to antifungals, right? So a strain that has the normal number of copies, you treat them with antifungals and they die. But one that has this extra copy, you treat it with antifungals and it lives perfectly well. So why is this happening? Is the sort of broader question. I want to point out one of these trisomies in particular. I won't go into detail about what these plots are showing, but basically here we're looking at a series of different Candida strains. There's 12 different strains here and a bunch of different strains here. And we're looking at all of their chromosomes. There's 8 chromosomes in the Candida albicans genome. What matters is, and this basically, these genomes have been sequenced. And when we see these dark bars that are highlighted in either purple or blue, that means there's an extra copy of that chromosome. And all of these populations, with the exception of this one at the top here, so the 12 below here, and then all of the ones here, these are all published studies, all of these are highly drug tolerant strains. So these are all strains that have been evolved to grow in one of the common antifungals, the azoles, and they are now quite resistant to the azole drugs. And you can see that they've acquired all of these different trisomies. But I think one sticks out kind of quite prominently, which is chromosome R. Chromosome R becomes trisomic in almost every single strain that is resistant to the azoles. So this really stuck out to us, right? So there's this one chromosome that keeps popping up in 3 copies, and we want to know why. Why is it resistant? And so what we the hypothesis of this work, or our assumption, is that this extra copy of a chromosome means that you get extra copies of all of the genes that are on that chromosome. And that means that you have extra sort of expression or dosage of all of those genes. And there's something about that extra dosage of the genes on that chromosome that's allowing survival. But we assume that it's not all of the genes. It's probably only a subset of genes. And so our question is, because there can be up to, there's hundreds, if not thousands, of genes on a chromosome. So our question is, which genes? Which ones are sort of driving this effect where you increase their abundance and they become more tolerant to antifungal drugs? And so to answer this question, our lab spent quite a while developing kind of a new CRISPR technique that allows us to get at this. I think people have become quite familiar with CRISPR for the most part, right? It's sort of a gene editing platform. We're using a modified version of that we call CRISPR, or not we didn't we developed this for fungi. We didn't develop it kind of in general. CRISPR activation, or CRISPR-A. And without going into too much detail, instead of a gene editing tool, it's a tool that allows us to increase the abundance of any of transcripts of any gene. So it basically drives high levels of transcription and expression from any gene of interest. And so because of that, we think that it allows us to sort of mimic the effect of this extra copy of a chromosome, but on a gene by gene level, because we can increase expression of each gene individually using CRISPR-A. And that's exactly the point, right? So instead of just targeting one gene, we can target, a second gene, a third gene, a fourth gene. We can target all these different genes individually and drive high levels of expression, which we think kind of mimics the effect of having a whole extra copy of the chromosome, but on a gene by gene level. And so this is work that was all, or is still sort of in progress, being pioneered by another PhD student in my lab, Nick Gervais. And so Nick basically set out to ask this question about chromosome R, right? So this chromosome that we know is, when present in 3 copies, makes cells very resistant to antifungal drugs. There's about 1,000 genes on chromosome R. And so Nick used this CRISPR activation system to overexpress each of the genes on chromosome R, 1,000 different genes. So he made these 1,000 different CRISPR mutants. And this yields exactly this. I'm showing only 4, but you can imagine this to be actually 1,000 different mutant strains, each one overexpressing a different gene on this chromosome. So it's 1 gene per mutant cell. And I'm just depicting these in different colors to sort of give this, to make this clear. And so the question is, from these 1,000 different mutants, each overexpressing a different gene, which of them are able to grow better in the presence of the antifungal drug? And so what we do is we take our pool of 1,000 different mutants and we grow it up. We grow it up in the absence of antifungal or with our azole antifungal drug. And what we expect is that some strains are going to grow badly, like, for instance, this red strain, which has disappeared from our population, but some of them are going to grow really well, like this blue and this green strain, which have actually increased in abundance in our population. And that's what we're trying to find, the ones that are growing better in antifungal drug when they have a particular gene overexpressed. And so that's, then the goal is to identify these new genes whose increased dosage results in antifungal drug resistance. And we use kind of next generation sequencing to do this. So again, we grow with and without drug, and we use sequencing to identify the relative abundance of all of our different strains in our population. And from the output of that, gets tons of different data looking at all these thousands of different mutants. But what we're really focused on are these few that are increasing in abundance. I won't go through exactly sort of what all of this means, but basically we're trying to hone in on a subset of genes that do better in drug when we overexpress them. And that's what we been able to kind of zoom in on. Whoops, I'm supposed to skip this one. Okay. And so from the screening, we were able to identify about 12 different genes that fit this criteria, right? That when you increase their dosage, you make them more resistant to antifungal. But this was in our sort of big bulk pool, and we want to assess these genes now individually. And so what we did then is measured a measure of antifungal tolerance called supra-MIC growth, which basically is just a measure of how well cells grow above sort of a minimum concentration of drug. And so basically it's a measure of how tolerant or resistant are they to the antifungal drug. And so that's what I'm showing now kind of on this graph. So we're looking at how tolerant all these different strains are compared to, this is our control strain. So our strain that has no genes overexpressed, it's sort of our wild type strain. And it has very low tolerance. These are 4 different strains that all have a trisomy of chromosome R, right? So we know these are tolerant, right? They are the ones that have the whole extra copy of the chromosome, and they are highly, highly tolerant to growth in drug. And now we're looking at our 12 different strains that we identified that each have a single gene being overexpressed. And what we find, excitingly, is that yes, indeed, they are a little bit more tolerant than the sort of control strain. So when you overexpress these genes, they are in fact a bit more tolerant to antifungal drugs. So that's cool. But what's surprising is that none of them have the same level of tolerance as the trisomic strains. And so what that tells us is there's probably some redundancy, or not redundancy, there's some um, sort of, we need multiple genes simultaneously in order to achieve this level of resistance. So it's probably not just one gene on its own, it's probably multiple. And so we decided to follow up on two genes. I know I'm highlighting three here, but two of them are kind of basically duplicates of each other. And so we picked these sort of two strains because they had by far the highest level of tolerance. So we figured these are probably the most important regulators of tolerance. And what we were pretty excited about when we identified these two genes is that, in fact, so these two genes are called SEC4 and YPT31. What's cool about them is, okay, there's a graphic missing. Oh, well, okay. What's cool about them is that they actually both perform the same function in the cell, or a related function. They're both involved in the vesicle secretory pathway, which is how cells kind of export vesicles to the cell membrane and push various things kind of out of the cell, and also remodel the membrane. And this was pretty cool because we screened 1,000 genes and we came down to these two genes, and it turns out they work in the same pathway. But we think that this pathway, the secretory pathway, actually has a really important role to play in antifungal tolerance, which we didn't know about previously. But the question is, do they work together, right? So without going on too deep a tangent, we developed a new CRISPR system that is going to allow us to look at overexpressing multiple genes at the same time, because we want to see if these genes are working together. And so that's what we did. And so we're looking now again at our measure of tolerance. Again, these are our control strains. They're not very tolerant to the antifungals. These are our trisomic strains. They're very tolerant to the antifungals. These are the two strains that overexpress these genes individually. So this one just overexpresses SEC4 and this just overexpresses YPT31, which are kind of medium tolerant. And then when we overexpress them together, sure enough, they are just as tolerant to the antifungal drug as the trisomic strains. So we think what this tells us is that indeed these two genes need to work together, and together they are in fact sufficient to induce this sort of level of tolerance or resistance to the azole antifungals, these secretory pathway mutants. And so we think this is pretty cool. The last kind of piece of data I'll show is that we also did kind of the opposite experiment. So instead of making the cells more tolerant by overexpressing these genes, we also took the tolerant cells, the ones that have the trisomies, these two gray ones, and we knocked down expression of these two genes in the trisomic strain. So instead of boosting expression in the regular strain, we're turning down expression in strains that have the trisomic chromosome. And sure enough, when we do this, when we knock down expression of these genes in the trisomic strains, these strains that were really tolerant before, they're not tolerant anymore. So we think that these two genes are kind of necessary and sufficient for this antifungal tolerance impact. And so, our model here looks now something like this, right? We've got our normal Candida albicans. It has two copies of chromosome R, and it's susceptible to drugs, right? It can be killed by azole antifungals. When it picks up a third copy of the chromosome R, when it becomes trisomic, it becomes highly tolerant, which I'm showing here in the kind of darker color, to the drugs. And when we take a cell that has two copies of the chromosome and we boost expression of these two secretory pathway genes, it is sufficient to make that cell tolerant to azoles. And then reciprocally, when we take the trisomic strain with three copies and we knock down expression of these two genes, well, that is sufficient to restore the sort of susceptible phenotype, to make them, again, sensitive to growth in drug. So we're pretty excited about this because, this is kind of a previously not understood process, the secretory pathway and its involvement in tolerance to these really important antifungal drugs. Okay, And so hopefully I have left lots of time for questions here, but the kind of main conclusions of the work we've been doing, we've been sort of validating and characterizing some first of their kind machine learning models that are allowing us to discover new antifungal drugs. We've been developing kind of next generation CRISPR techniques that allow us to alter the levels of gene expression in a cell that we is allowing us on a sort of large scale to characterize some of the genetic determinants of antifungal drug resistance. And then we're using some of these sort of big pooled mutant libraries, like I showed, to uncover some new regulators of trisomy mediated antifungal tolerance and drug resistance. And so I'll just highlight as we close that I think I sort of said the word we a lot. None of this work is done by me. I sit in my office. All of this work is done by really awesome, exceptional grad students, postdocs, and undergrads as well in the lab who contributed to this work. And I think I showed some of Nick's work, which was the trisomy. Mia and Eileen, who's not shown here, have done some of the machine learning work as well. And so it's always like a big kind of collaborative effort. And so, yeah, with that, I want to thank you all so much for listening. And I'm excited to hear what questions you have.
Thank you. That was amazing. I feel like we have one question so far in the chat. This is from Christopher. Trisomy is often terrible for an organism. How do these fungi tolerate trisomy, and can the trisomy be attacked?
Yeah, that's a great question. Should I leave my slides up or should I like,
Sure,
do
you
you know what
can
I
do.
mean?
Might
Or should I
be
go?
people
I'll,
to
I'll,
print.
I'll stop sharing so you can see me. And if I need slides, I will start sharing again. Okay. Yeah, that's a great question. And it's true. Like, I think from whatever grade 10 biology or whenever you start learning about this on, the kind of canonical principle you learn is that the mission of the cell is to, with high fidelity, replicate its genome, right? So, and to properly divide so that the right number of chromosomes have been, segregated into each kind of subsequent cell. And yeah, and what we know in sort of humans, for sure, is that when the wrong number of chromosomes is found, it tends to be very detrimental to the cell and to be a big problem. And fungi really turn this sort of on its head because that doesn't seem to be the case. And all this work is done in Canada, but we see this across the fungal kingdom. That we often see these like inappropriate number aneuploid is what we call it, chromosomes, where it will, I guess it's sort of a balance where these aneuploidies can be very detrimental to the cell in some circumstances, but can be very beneficial in other circumstances. So the chromosome R trisomy, for instance, that we study, those cells grow really well in antifungal drug. They actually grow quite poorly without antifungal drug. And so there's a trade-off. And so there is, in fact, a fitness cost to this trisomy, but only in some circumstances. In other circumstances, it's actually beneficial. So it's complicated, to say the least, and it is something that people are trying to understand because it's not sort of the expected outcome of having this sort of very dynamic changes in genome. And to the question of like, can it be attacked, I think is certainly a really interesting one. It's difficult because, yeah, anyways, yes, it is. It's an interesting question for sure. One of the things that we are also looking at in the lab is trying to understand what some of the genetic determinants of aneuploidy are. So not just the genetic determinants of the resistance, which is a result of aneuploidy, but actually like what are the genes that control the propensity for cells to become aneuploid in the first place. And that is something that we're trying to understand because maybe that then is kind of an interesting target, something that you can inhibit and kind of lessen the frequency of these things.
Go ahead. I see we have a hand raised. Feel free to come off mute and ask any of your questions.
Hello, Dr. Shapiro. One of our participants has a question. His name is introduce your name and
Yeah, I'm Akshat, and I was just wondering, so if I'm reading it correct, like azoles were one of the three types of antifungal like treatments. What were the other two?
Yeah. Yeah. So azoles are one of the most common antifungal drugs and are also the most common antifungal that's like sprayed on crops. In the clinic, the other sort of major first line antifungal are the echinocandins. They target the cell wall of the fungus. The azoles target ergosterol, which is in the cell membrane of the fungus. And then there's a third category of antifungal, the paulines, which actually also target the membrane, but in a different manner. Those are the major classes of antifungals. There's sort of another class that is sometimes used, but the rate of resistance is so high that it's never actually used on its own as an antifungal. It's only ever used in combinations with others because of how quickly resistance happens.
Okay, thank you.
Thank you for your question. We have a couple of other questions here in the chat. Another one coming in, having gained some insight into the function of the genes that confer tolerance to the azoles, I believe, does that provide a pathway for research into compounds that could attack that functionality?
Yeah, that's also a great question. And that's certainly something that we think about. I mean, we start at this sort of fundamental question of like, how is this happening? Like, what is happening in the genome? But obviously, the hope is that, probably many steps downstream, but that at some point, maybe this allows us to better understand ways that we can prevent this from happening. Um, in the case of this sort of secretory pathway, which we've identified, we don't yet quite understand how it's linked to antifungal tolerance. What we suspect is possibly the antifungal is you actually gets into vesicles and then gets pushed out of the cell that way. And so if you increase the rate at which that's happening, you're basically pumping out the antifungal more quickly, which would allow them to be more tolerant to the antifungal because you're just like pushing it out of the cell. And so, yeah, thinking about a way in which you could like inhibit that process could potentially be really impactful. Making, developing drugs is really the hard part. So that's, it's identifying the pathways is sort of step one.
They're all really great questions. All right, we have another one from Carol. Are the antifungals that are developed for, say, the Candida fungi also important for treating Aspergillus fumigatus? Related,
Yeah.
how does tolerance to an antifungal for a human figure into drug development?
Yeah. Okay. So part one, yes. I mean the hope with the development of any antifungals, like the ones that I talked about that we've been discovering, the machine learning ones, I didn't show data for Aspergillus, but we did test them against Aspergillus, and they are effective against Aspergillus fumigatus. Um, Aspergillus is very different from Candida because it's a mold and it grows incredibly differently. It infects different sites of the body. It's different in many ways. But evolutionarily, Candida and Aspergillus are not like too dissimilar from each other. So while there's certainly some drugs that work against one but not the other, the hope certainly is always to develop drugs that work kind of as effectively against all or as many fungi as possible. And so, yeah, that's definitely something that we kind of have been thinking about and testing. The second part was about tolerance. What was the?
Related
I can pull
to
this thing.
how does tolerance to an antifungal for a human figure into drug development?
Yeah, so we never know. Certainly, a goal with any sort of new antifungal drug is that will you will have the sort of lowest rate of resistance or tolerance as possible. That being said, it can be really hard to predict, right? So what we often do in the lab, and we do this like when we discover new antifungals, like with our machine learning, is we try to see how quickly resistance can develop. So we, take our cells and we hit them with the drug and we passage them, and we do this like we basically, do like in vitro experimental evolution to see how quickly they develop resistance. And obviously, the hope is to find something that doesn't develop resistance or tolerance very quickly. But it can also be really hard to know because what happens in the body is just like totally different than what happens in the lab, surprisingly. Um, And so, and especially because fungi have this crazy ability to do things like retain entire extra copies of chromosomes, um, which is a really dramatic change to the cell and can very rapidly shift kind of how resistant they are to something. Um, it's sometimes can be hard to know what's going to happen, but yeah, it's all we try, to take this into account when drugs are being developed.
We have a hand raise from our SSP biochemistry group.
Hi, my name is Austin. I kind of had a two-part question related to the machine learning aspects. So, the first one is, did you see any patterns or correlation between when you started with the generative and moved on to like the predicting its effectiveness, like those two models? Like, did they agree? Like, were the most promising ones actually shown to be effective? And also, is there a concern that like due to this two-step ML process that you're not actually testing these molecules? How effective is this ML process when you're not testing these initially and you're mostly staying with ML?
Yeah. Okay. So the first question is, how well do the two models agree? Was that
Mhm.
sort of right? So
Yeah.
we're only like just getting the data of the generative model, and it uses the discriminative, like the training data and the previous model as part of it. So I mean, anyways, we don't. The short answer is we don't like yet know because we basically haven't like tested those molecules yet. Yeah, so we try, and for the second part, we try to get to model, or sorry, to compound testing as early as kind of realistically possible. Some of this comes down to cost and scope, right? Like, because yeah, we end up generating thousands of potential, like, predicted antifungals. We just can't realistically like buy and test all of those. It would just be insane. And there are things that we want to try to rule out, like toxicity, et cetera. So once we've, performed some kinds of quality control, we try to sort of get to the actual like testing stage as soon as possible. And that then gives us more data that we can sort of like keep feeding back. And once we have tested like in lab molecules, we can keep feeding that back in, but those numbers are just much smaller than our kind of initial screening.
Thank you so much.
Great. Okay, I think we have time for one last question. If anyone wants to get one last question out before we wrap. Okay. Well, thank you all so much for being here for today's session. And thank you so much again, Rebecca, for sharing with us. This was such a really invigorating and really engaging session. So thank you all. This was amazing. You can find this recording of this session on our YouTube channel, SSP International, the Summer Science Program. You can also find it on the SSPI network. I will also plug our next Curiosity Lab, which is August 9th from 3 to 4 p.m. That is the Robotic Telescopes Real Data, How a Global Observatory Network Powers Education, Citizen Science, and Space Domain Awareness. And that's going to be with our speaker, Emma Loudon. So if you guys are interested, please go to our website and you can register for that one. Thank you all so much again for being here with us. And we really appreciate you. Enjoy the rest of your Sunday.
Thanks, everyone. Great questions. Thank you.
Thank you.